As a result of world-class expertise in algal research, Cawthron scientists have developed a method to commercially grow a New Zealand alga, then extract and purify a compound from that called neosaxitoxin. It's a critical ingredient in a novel and groundbreaking medication for pain.
Neosaxitoxin is a so-called sodium channel blocker. It's a compound produced by algae that can make us very sick, but used in the right way, we can actually use it as a painkiller, a highly efficient painkiller. So we're trying to develop it as an opioid alternative for the treatment of post-operative pain. The opioid family, to which morphine belongs, for example, is a class of compound. They're very efficient painkillers, but they come with a range of side effects like constipation, you get tired, and then there's all these challenges with addiction. Neosaxitoxin is a single dose treatment that will give you pain relief for up to 3 days, and it doesn't come with any of these side effects. So it's very suitable for children, for example. So yeah, it's a very cool compound. Neosaxitoxin has been known for quite a while. People know it's a highly efficient painkiller, so it has been developed by a Chilean pharmaceutical company called Proteus, together with Harvard in the US. And they've taken it through phase 1 clinical trials, and it works really well. The challenge with neosaxitoxin is that it's a very complex little chemical. You can't readily synthesize it in the lab, and it has been proven very difficult to isolate it from the natural producer. And that's our role in the project, is to generate a supply of a high purity compound for clinical development. We have been working for nearly 30 years on seafood safety, and here in New Zealand that relates to detection of these type of paralytic shellfish toxins in food. So we know toxins really well. We produce these compounds for analytical standards and sell them all over the world, so we have a good knowledge on toxins. So we were approached a few years back by a European partner who wanted larger amounts of compound for studies, and we provided that to them, and they got back and said, "Ooh, I've never seen it this pure before. Can you make much more?" And we could. And then we got involved in the actual drug development. So our role is to make neosaxitoxin in large amounts of a high purity. We envisage that this will be used by hospitals all over the world, and we think that the initial global demand would be around 12.5 million doses, and that's only 500 grams of the compound. One of the major advantages with neosaxitoxin as a compound is that it's water soluble, and that means in the body it won't cross the blood-brain barrier, so it doesn't have a central effect on your body. So you would use it for localized pain treatment. The enabling factor for us was our access to our microalgae collection. So in here we found a native strain of microalgae that was a high producer, a particularly high producer. And we can now grow that at 1,000 times higher density than it's found in the wild. The algae is called Alexandrium pacificum, and it's a dinoflagellate from the ocean.
The kind of volumes we're talking about at this algal centre, so this is, I guess, a pilot plant. It's kind of just before commercial scale. Initially, we start with a 100 ml flask of algae, and we bulk that up to about 2,500 litres. Our larger bioreactors here are 1250 litres, and we've got 2 of them. We also have a 100 litre smaller bioreactor that we use as a stepping stone, and then we have quite a complex system of 1 litre bioreactors that we use for optimisation and scale up from those initial 100 ml flask volumes. We control a lot of the parameters for the algae, and we have, I would say, reasonably broad tolerances. Light is very crucial, as with the timing of the light and dark cycles and the nutrient composition, so small deviations from that can really dramatically affect how the algae grows. From biomass, which we harvest from our bioreactors, we dewater, so we remove all of the seawater and just have an algal sludge, kind of like a paste, if you will, a little bit of similar in texture once all the water's removed to Marmite. We heat that up and basically cook the compound of interest out of the cells, and then we do a step called cross-flow filtration, which is similar to how they do dialysis in the hospital, and that's how we remove all of the algal particles and get our crude extract. This is then transferred to our lab in town, and then there's a series of purification and chromatography steps that isolate and purify our compound of interest. And finally, there's a chemical conversion step which converts it into an active pharmaceutical ingredient. That heads off overseas for formulation and then for clinical trials at the moment. Cawthron does own the IP, and we've patented the techniques for the chemical conversion and the purification. Our strain is a very, very good producer, and that's our house strain as well. So we feel quite comfortable sharing this information. But in the future, what we're looking at is scale up. So this is just the pilot scale at the beginning. We'd expect to sort of go 10 times or maybe slightly larger than this. We can now produce around 1 gram of this compound a week. So in a single gram, a fifth of a teaspoon, if you will, there's 25,000 doses.
We are trying to organize phase 2 clinical trials, and we need to team up with a commercial partner for that. So the compound needs to make it through phase 2 and phase 3 before we can find it in clinical use, and that's a few years down the track.